The human central nervous system is composed of delicate, soft tissue. Without external support, the brain is incredibly vulnerable to its own physical mass. In open air, a human brain weighs 1,400 grams. Inside the fluid-filled skull, it weighs just 50 grams. Without this fluid, the soft tissue would sink against the cranium floor. Gravity would compress lower neural structures, cutting critical blood vessels. To prevent this structural collapse, the brain relies on 3 coordinated lines of defense: the bony enclosure of the cranium, 3 membranous layers called the meninges, and a bath of cerebral spinal fluid. The neurocranium functions as a dynamic, fluid-filled suspension system. This physical architecture is what allows the central nervous system to survive its own anatomical density. The brain and spinal cord are tightly invested by 3 concentric membranes: the innermost pia mater, the middle arachnoid mater, and the outermost dura mater. Clinicians group these layers based on their structural properties. The inner 2 layers, the thin pia and arachnoid, are together known as the leptomeninges. The tough, thick outer layer, the dura mater, is classified as the pachymeninx. This cross-section illustrates the exact sequence of these layers. Moving inward from the scalp and skull bone, we encounter the dense dura mater, followed by the arachnoid mater, the subarachnoid space, the pia mater, and finally the folded cerebral cortex. Memorizing this specific architectural sequence is a mandatory prerequisite for diagnosing head trauma, tracing hemorrhages, and navigating any invasive cranial procedure. To understand how these layers interact, we construct them from the inside out, starting at the exposed surface of the brain and adding the innermost meninx, the pia mater. The pia is extremely thin and highly adherent. It acts as a biological shrink wrap, closely following every raised gyrus and dipping deeply into every indented sulcus across the brain's surface. It is also highly vascularized. Folds of the pia mater enclose tufts of capillaries to form the tela choroidea. When lined by specialized secretory ependymal cells, this structure forms the choroid plexus, the exact site where cerebrospinal fluid is actively secreted. The pia mater operates as the ultimate metabolic and vascular interface. It regulates the immediate boundary between delicate nervous tissue and the bulk fluids circulating outside it. Immediately outside the pia is the arachnoid mater, named for its translucent, cobweb-like appearance. Unlike the tightly bound pia, the arachnoid loosely envelops the brain. It bridges across the sulci and surface irregularities rather than dipping down into them. Because the pia follows the deep grooves while the arachnoid spans across them, a distinct gap opens up between the 2 membranes. This gap is the subarachnoid space. This space is filled with circulating cerebrospinal fluid, creating the primary shock-absorbing water cushion for the central nervous system. The volume of this space varies. Around the cerebral hemispheres, the gap is narrow, but at the base of the brain, the arachnoid bridges over large surface depressions, creating widened pools of fluid called subarachnoid cisterns. Within the healthy cranial meninges, the subarachnoid space is the only actual physiological space. It provides the physical pathway for CSF circulation and houses the neurovascular vessels supplying the brain. Capping the entire system is the outermost, thickest, and toughest membrane, the dura mater. This dissection image demonstrates the physical reality of the dura mater. It is an opaque, fibrous sheet lying directly beneath the skull cap, physically sealing off the underlying brain and leptomeninges. The cranial dura is composed of 2 distinct layers: an outer endosteal layer, serving as the skull's periosteum, and an inner meningeal layer. These heavily fused layers intentionally separate to enclose cranial venous sinuses, establishing the main venous drainage channels for the brain. Beyond enclosing the sinuses, the inner meningeal layer of the dura folds deeply inward upon itself, partitioning the open cranial cavity into highly specific compartments. These folds include the falx cerebri, the tentorium cerebelli, the falx cerebelli, and the diaphragma cellae. This dual-layered architecture serves 2 distinct mechanical purposes. It houses the low-pressure venous drainage network, and it physically locks the distinct lobes of the brain tightly in place, minimizing rotational shearing forces during physical trauma. The entire system relies on a precise hydrodynamic cycle. At any given moment, there is roughly 150 milliliters of CSF in the system, continuously secreted by the choroid plexuses located deep within the brain's ventricles. From the lateral ventricles, the fluid flows through the interventricular foramina into the third ventricle, passes down into the fourth ventricle, and finally exits outward into the subarachnoid space. Once in the subarachnoid space, the fluid circulates continuously, moving upward over the cerebral hemispheres toward the top of the skull. This diagram illustrates absorption. Fluid at the top of the brain is pushed through arachnoid villi, which grow into arachnoid granulations. These granulations pierce the meningeal dura mater, acting as one-way valves that dump CSF directly into the superior sagittal sinus. This continuous loop of deep ventricular formation and superficial venous absorption tightly regulates intracranial pressure. Any blockage along this pathway, whether from a tumor, inflammation, or hemorrhage, results in an immediate, life-threatening rise in pressure. Understanding fluid dynamics also requires differentiating actual anatomical spaces from potential ones. In a healthy cranium, the epidural space outside the dura and the subdural space beneath it are entirely collapsed. They are potential spaces. Their clinical danger arises during trauma. Arterial or venous bleeding can force these layers apart, creating vast pockets of blood. Because the skull cannot expand, these epidural or subdural hematomas rapidly compress the brain tissue inward. We can also apply this anatomical logic downward, tracing the meninges as they extend past the base of the skull and down the vertebral column. The solid spinal cord ends in the upper lumbar region, but the subarachnoid space, along with the dura and arachnoid layers, continues further down, creating a wide, CSF-filled pocket known as the lumbar cistern. This structural difference provides a safe window for clinical intervention. By inserting a needle into the interspace between the third and fourth lumbar vertebrae, a clinician can access the lumbar cistern and draw a sample of cerebral spinal fluid without risking damage to the solid tissue of the spinal cord. The central nervous system is heavily guarded, but its defenses follow a strict structural logic. By mastering the sequence of the meninges and the fluid dynamics of the subarachnoid space, clinicians gain the precise physical coordinates required to access, diagnose, and decompress the human brain and spinal cord.